Good afternoon, everyone. I am Claudia Marino, a postdoctoral fellow in Dr. Arboleda Velasquez Laboratory at the Department of Ophthalmology, Harvard Medical School. Thank you all for attending today's MAPS round. It's my great honor to introduce Dr. Simone Dujardin. Dr. Dujardin trained as an engineer in biotechnology in Paris, France, and then joined the neurobiology PhD program for the University of Lille, where he studied the mechanism of tau cell-to-cell transfer in the lab of Dr. Luc Bois and Dr. Marvin Collin. Simone then moved to Mass General Hospital in the lab of Dr. Bradley Hyman, first as a postdoctoral fellow and then as an instructor in neurology, where he continued his seminal works on tau propagation with a focus on the mechanism of tau seeding. The title of his talk for today is Tau Propagation in Alzheimer's Disease, Mechanism and Disease Relevance. This meeting, as you heard, will be recorded, so please feel free to ask any question you might have at the end of the talk in the Q&A session, so we can let Dr. Dujardin answer at the end of his talk. Please join me welcoming Dr. Dujardin.
Thank you very much. Thank you for having me. It's a great opportunity for me. So as Claudia just said, I'm going to talk about tau propagation. So that's something I've been working on for 10 plus years. And I actually recently left the Mass General Hospital, but all the work I'm going to present today has been done in the lab of Dr. Bradley Hyman at Mass General Hospital. So, yeah. So basically today I'm going to give a quick introduction and then talk about tau cell-to-cell transfer and then tau seeding. I'm going to touch base a little bit on different strains and how biochemistry can help us differentiate these strains in Alzheimer's disease. And finally, if we have time, I'm going to talk about a last project that we've been doing on the resistance of these seeds to treatment. So as you all know, Alzheimer's disease is characterized by different histopathological findings in the brain. And the most known of them are probably the appearance of amyloid beta plaques outside the cells, and the presence of neurofibrillary tangles, which were here drawn by Alzheimer's stem cells in these first papers. And these neurofibrillary tangles were actually shown in 1986 to be full of a protein called tau that aggregates inside neurons and form this flame-shaped kind of beautiful findings in the brain. So these neurofibrillary tangles in Alzheimer's disease very interestingly progress over years in the brain, and especially it was shown by Brack and Brack in 1991 and followed by others that deposit in a very stereotypical way. What I mean by that is that it always starts in the same place. Usually people are saying that it starts in the entorhinal cortex, and then it spreads from the entorhinal cortex to connected regions such as the hippocampus, and then more regions, and so on and so on. And it takes years to happen, and that's accompanied by loss of synapse, a lot of neuroinflammation, and ultimately loss of neuronal cells. And this progression, this stereotypical staging progression of tau in the brain correlates very well with symptoms in Alzheimer's disease. And interestingly, tau aggregates in not only Alzheimer's disease, but around 20 other pathologies, which are known as tauopathies. And in a number of them are very, very rare, so it's very difficult to do staging, but some of them are not that rare. And this kind of staging of tau pathology appearance have been shown in at least 3 others this pathology, which the stagings were different, so they didn't progress the same way, but you could still see this stereotypical progression from connected regions to other connected regions. And so that's the case, for instance, in the quite known progressive supranuclear palsy, alveolar brain disease, or Hspgs disease. And this has led to a hypothesis that tau could actually propagate, such as a prion. So what for just sake of a short introduction, prion were actually described in 1992 by Stanley Persiner, and why we think that tau and other proteins are prions is that prions are defined by 2 things. The first thing is that it's transmissible. So what I mean by that is that you can be in contact with a prion, and after a couple of time, well, like some sort of time, you're going to develop the disease, basically. So that's transmissible. And then the second thing is that they are in terms of biochemical feature, you have strains of prions. So they are defined by structural differences, but also by a whole bunch of biochemical characterization. I'm going to touch base on some of them that we perform for tau. And so basically, people were trying to understand if other diseases of the brain were similar to prion in some way. And we had some hints in the early, like 2008, 2009, that it was actually the case, at least for 2 of these other misfolded proteins in the brain, alpha-synuclein and tau, and also amyloid beta, actually. So what happened with alpha-synuclein is that some people did some graft of neurons into a diseased brain, and the neurons were totally fine. But after a couple of years, they could see actually Lewy body coming inside these neurons, showing that transmission has happened between the diseased tissues to the actual graft, which was the first kind of like experiment showing that, oh wow, that might happen. And then people have done work in mice where they were basically taking a diseased mice full of pathology and injecting that injecting a brain lysate into a naive young mouse and seeing basically a pathologic conversion of this mouse. And that has been done both with amyloid beta and tau. So it seems that first analogy with prion disease, tau pathology is transmissible. And the second is that indeed you could have hints that there are some strains of tau existing, and that slide I've made like a couple of years back. And the first evidence that we had that tau could happen in different strains were basically the shapes of the lesions in the brains. So different diseases had different shapes of pathology. And so these are the neurofibrillary tangles, but you also had some pathologies with some glial lesions, and so such as an astrocyte or oligodendrocyte. But even within neurons, you could see very different sort of morphology, with here peak bodies that don't look alike at all from neurofibrillary tangles, or arteriophilic grains here that are really punctured, and were pretty like typical of certain diseases compared to others. And the actual definitive proof that strains existed were demonstrated in the last like 2-3 years by the use of cryo-EM, which was a real breakthrough for the field. showing that actually when you take aggregates from tau from different diseases, you have very different structures of these aggregates, showing that definitely you have structural strains happening that define different diseases, and that was a breakthrough from a team in Cambridge University in UK. And so, basically, the questions we had from the real beginning was like, why such staging? Can it be from vulnerability of cerebral networks, or is it really because of an active prion-like spreading? And when I say prion-like spreading, again, I'm talking about a transfer, an active transfer from misfolded species from one neuron to another neuron. And in the second neuron, you have a recruitment of naive tau inside misfolded aggregates in a mechanism that is called seeding. So represented here. And so we wondered that, and then subsequently we had like many questions. So what are the mechanisms of progression from region to region? Is this, is what we see in vivo or in cells is relevant to anything in the actual disease? Are there any strains within disease in addition to strains across diseases? And then can we do something about it? And so when I started working on it in 2010, the first questions we had, does tau actually transfer from neuron to neuron? And that was not an obvious question at that time, because tau is a cytosolic protein, so it's not anywhere secretory pathway. So just the fact that it would leave a neuron was at that time not something we would take for granted. And so we developed a model of rats that basically was an injection of lentiviral vectors inside the brain, and then we could evidence transfer of tau from the injection site to different regions, very, very distant, from, like olfactory areas or subiculum or things like that. And so that was quite the first evidence. We did like a bunch of controls to verify that it was actually legit. And that was also confirmed by many teams actually working on the same thing at the same time, and in different type of models. Some of them were transgenic models with an exclusive expression in the internal cortex, and some of them were also models of injections, but not in rats and mice. But basically, bottom line is yes, tau is transferring from cell to cell. And then there was a bunch of mechanistic studies to understand how it happened. I don't think we have a definitive answer on that, but there are probably a lot of different mechanisms involved, including vesicles, or so like a free floating seeds. So you get the first part of it, and the second part is the seeding part. So do tau, misfolded tau recruits naive tau inside these aggregates, and does this aggregate basically propagate that way? And a lot of work has been done in 2014 to 2020, basically on that, showing that yes, it does happen. Yes, you can show that in different. So all of these papers are basically proof of that. So you can have aggregates coming from human or in vitro generated, or coming from transgenic mouse models, and they all recruit different tau in vivo or in vitro, or even fully in tubes. So seeding is a process that also happens. So I'm basically done with the introduction. Now I'm going to go to more like data that we generated first on the cell-to-cell transfer. and then more on the tau seeding in the second part. So once we had demonstrated that tau was basically moving from cell to cell, the advantage of viral vectors is that you can really ask a bunch of questions. So we asked the questions of different tau isoforms in another paper that I'm not going to present today. And there are differences within the different isoforms, but another question we had is like, do differences in genetic background actually influence the propensity of tau to move from cell to cell? And to answer this question, we basically use this system that is an injection of AAVs inside the hippocampus or the anterior cortex of mice. What these AAVs are interesting because they express GFP and a tau protein, but in the middle of that, you have a small peptide. It's called the 2A peptide, and it is self-cleaving. So what will happen is that in the cells, you have basically GFP and tau expressed, but they are separated. So you can really see using the GFP what cells were transduced, and then you can follow the tau protein if it's moving or not. And so when you look at images, for instance, so you have here a mouse that was injected in the hippocampus, so here with this construct, and here in the enterocortex. So one injection on one side in the enterocortex and the other side and the hippocampus. And when you look at a closer look at the enterocortex injected side and you do a closer look here, you can see neurons that are only tau positive, which is basically a sign that it's not a neuron that was transduced by the AAV because it doesn't have the GFP signal, but it has human tau. And because we added human tau only with this AAV, this means that these are the recipient of tau propagation. And so what we did is that first we tried to compare young versus old mice, showing that old mice have more propagation than young mice. And then we took mice with very diverse genetic background and we injected that and we quantified the amount of propagation in the different strains. And what you can see in this graph here is that some strains have a lot of propagation and some strains are very resilient to it. So tau actually doesn't move that much. We compared male versus female and we don't really have a difference. And then we also looked at tau pathology and we didn't have a clear difference in tau pathology for the different strains. So basically here, we confirmed that in another study that is ongoing right now with different models, but that's really showing that the genetic background of mice and I guess maybe humans as well are really important and into this cell-to-cell transfer system. But of course, on this particular cohort, it was not possible to pinpoint actual genetic factors that will actually lead to that. So future studies really need to address that. So let's go to the second part of my talk. So for this one, we basically used a system that quantified tau seeding from basically any source. So what is the system is that we have human brain lysate here from AD patients that we use frontal cortex. And if you homogenize that, you get a brain lysate. And if you put that on top of these cells, so these are hex cells that express tau. So that's a truncated form of tau fused with CFP and YFP. They don't really aggregate, so there is no transfer from CFP to IFP in general. But if you put seeds from any source basically on top of them, they will aggregate, and they will have some Fred signals. So basically energy transfer from the CFP to the IFP, and you can quantify that using flow cytometry. So here, for instance, you have a population of cells that have some Fred positive signal compared to a control that almost don't have any Fred positive cells. And you can also see that using basic imaging, where you see this punctures here, these like aggregates forming inside the cells. And so our idea was to test a bunch of cases from our brain bank. And so we took like, we wanted to have at that time a homogeneous cohort. So we took, 80 cases, frontal cortex. They were all BRAC5 or 6. They were all like seen at MGH and confirmed in autopsy at MGH. So we're pretty certain that this cohort would be quite homogeneous. And when we were sitting on that, that was quite our surprise that it was actually very heterogeneous, with some cases was very, very high seeding, and some cases was fairly low seeding. So we were a bit confused for a moment, like, okay, our assay might be very variable or things like that. So it's not an assay problem, because then we confirmed this data using different assays. So especially a seeding assay using primary neurons, where you basically see after 2 weeks with incubated with brain homogenate, you can see basically neurofibrillary tangles forming inside the cells. So these cases here were very much in line with what we saw basically in the second assay. And furthermore, we did an in vivo assay using a transgenic mouse model of Alzheimer's disease. Well, yeah, not really Alzheimer's, transgenic mouse model of tauopathy that developed tau pathology around 6 months of age. So what we did was to inject earlier than that, so at 2 months, and sack the animals around 4 months before they were supposed to develop pathology. And then this 2 month period was enough to induce seeding, and you see that's what you can see here. So on a low seeder, you don't see any neurofibrillary tangles, but you see them coming for the high seeders, and that was basically quantified here, following very well or two other assays. So there is a high seeding heterogeneity basically from samples on this cohort. And as I said, we're a bit confused, but then we decided to look a bit further, and all of these cases were seen at MGH by neurologists and have been they've been seen in the clinic, and in the clinic they do a bunch of things, but one of the things they are looking is the CDR, so the clinical dementia rating. And a person that is cognitively normal would have a CDR of 0, and a person that is fully demented will have a CDR of 18. And so here what you see is let's say a classical, I don't like this term, but a classical AD patient who comes for his first visit 6 years after his disease onset, and then has a CDR taken every year, and the CDR slowly progresses and then reaches the maximum. So that's like one patient. But when you look at like, so the first cohort has 32 patients. So the 32 patients are plotted here actually. And so actually that's where you see that there is a high degree of also clinical heterogeneity in AD. With patients that basically come like a year after onset, rather than some patients come like 10 years after onset. And some patients will progress very, very fast, and some patients will never even like reach any like CDR-18. So we decided to correlate basically this rate of AD progression with tau seeding, and also the ages of onset. And what you can see in these two graphs here is that we have a correlation, significant correlation between tau seeding and the rate of disease progression. And you have an inverse correlation between the age of onset and tau seeding in these cases. So what does that mean? Is like the younger the patient was at onset, the higher the seeding was, and the fastest the disease progression, the highest the seeding. That led us to think like, what makes this patient different? Why a patient would have a high seeding versus the other a low seeding? Would it, you know, like what happened? And that's where we decided to go for a full biochemical characterization of these patients to understand what is the molecular characteristics of these seeds and what differentiate one seed from a high seeder versus a low seeder. And so the first thing that came in mind is like, okay, maybe just the high seeder has more tau. You know, if you have more tau, you have more seeds. If you have more seeds, you have more seeding. But actually, it was not the case. Like when you looked at total tau, so these are the same patients. So they are ranked from low seeders to high seeders, and that's going to be the same for the next couple of slides. Low seeders being in blue, high seeders being in red, and patients going from 1 to 32. So there was not really a correlation between the amount of total tau and the seeding. When we looked although at an assay for that quantified the amount of oligomeric tau, already we could see a positive, you know, a significant correlation between tau seeding and the amount of oligomeric tau. And we wanted to go a bit further with that by using, you know, by trying to purify these oligomeric tau species and quantify them. And the first thing we did was to put them in a size exclusion chromatography. So we put this brain lysate, and we basically got fractions of high molecular weight versus low molecular weight tau. So the multimeric tau will fall in high molecular weight, and the monomeric and truncated forms of tau will fall into the low molecular weight. And we analyzed that using SDDH as a first, and you can see that you have difference in terms of amount of this high molecular weight. So this here is the high molecular weight fractions, and here is the low molecular weight fractions. So you can see they have difference from case to case there. But what was interesting is we put that back into this alpha ELISA to understand where was tau. And if you do a total tau, you can see that basically most of the tau falls around like you know, 10 to 15, and that's where it's expected to be, because that's basically the molecular weight of the monomeric tau. But you have a slight peak here between you know, fraction 2, 3, 4, which was interesting. And already you could see that the high seeders have had a bit more. And when you do oligomeric tau, you can basically confirm that this oligomeric tau falls here. And you can see that the high seeders patients have way more of this oligomeric tau inside this fraction. Then we looked at different phosphosites, showing that this high molecular weight fractions of tau were hyperphosphorylated basically. And we ran seeding on these fractions too, showing that most of the seedings were actually present in these fractions of high molecular weight as opposed to monomeric tau. So what that means is that, first of all, these patients have more seeding competent high molecular weight oligomers yeah, basically. And then we went further to that. We wanted to know if their content into post-translational modifications were different from patient to patient. And so we purified these high molecular weight tau, and we sent them to Professor Dujardin at Harvard Medical School to be around a mass spectrometer basically, and find out if they were phosphorylated and at what side and how much of this phosphorus. So bottom line, and that was very interesting, we could see a signature of phosphorylation in these oligomers, and that signature was correlated with the amount of tau seeding. So I'm not going to enter into the details. Everything is published, so if you want to see, you can go. We continued a bit this study, and that's something that is in revision right now with Professor Dujardin, by doing basically a signature of all PTMs in addition to the phosphoepitopes. And these species are also, in addition to be phosphorylated, they're also ubiquitinated and acetylated, and all of it quite tightly correlate with seeding. And when you do ubiquitin immunodepletion, you can see that you're reducing drastically the amount of seeding. And I invite you actually to look at a paper that Judith published last year, actually this year in Cell, where she compares different tau species, and especially our high molecular weight tau species, where she can basically, as I said, find a signature of these post-translational modifications on this tau species. So we basically then wondered if structurally we had a difference between the different patients. And one first experiment that we did was basically to look at these seeds happening in live imaging and see if we could find out some differences by doing that. And that's what I did with a very talented undergrad student at that time that is now in a PhD program at HMS actually. Well, the MIT and Harvard program. And so what you can see here is that when you put seeds on these cells and you image them, you have this very typical curve here that includes a small lag phase, a quite rapid growth phase, and a plateau of after 35 hours somehow. And also you can basically quantify, in addition to that, the shapes of these aggregates. And so that's what we did. And when you take the same sample and you vary concentration, so that's exactly the same sample, just varying concentration, you can see that the lag phase and the plateau phase are really in line with the amount that you put at the very beginning. But interestingly, the slope, so the growth, is quite stable. So it doesn't correlate at all with the amount that you put at the beginning. And on another hand, if you take the different 80 cases and you try to find if you have differences between lag and plateau and a growth phase, you can see that you have differences into the growth phase, and that these differences in the growth phase correlate quite tightly with the aggregate shapes, basically. And for us, this is an indication that you have structural diversity in the seeds from patients to patients. And that's something we also saw by using a protease, so proteinase K. And so we did basically digestion of this high molecular weight tau using different concentration of proteinase K. And what you can see here is that you have different degradation products that are more resistant in some cases than other cases. And that is significantly different in high versus moderate or low seizures, clearly showing that you have structural differences between patients. So do you have time? Yeah, I do. So the last bit of information is about how resistant these seeds are. And what led to this project is that basically at the very beginning of this old tau propagation story, we had questions about, okay, we're talking about prion-like, but prion is, you know, everybody kind of freaks out when you talk about prion because it's highly transmissible. You really don't want to have that hanging around the lab and things like that. And so a lot of people have raised questions, neuropathology lab and things like that. So first questions, are these brain homogeneity infectious or transmissible or even, as I said, prions? And if yes, can you deactivate them by using some sort of treatment or autoclaving, for instance? And so that's what we asked here. So that work has been done with a technician that worked with me for a couple of years, and Elise Fernandez, really bright technician. And so basically what we did is we took some brain homogenate from different patients and we applied different treatments. And the first things we did was just to boil it and filter it, like filter to like a regular lab filter at 0.22 micron. Doesn't change the seeding capacity of the solution. Boiling autoclaving though worked quite well as a first blush. We also tried to vary the pH, so have an acidic pH or have a basic pH, and that didn't remove seeding ability to the samples. Fixation using paraformaldehyde worked, and formic acid, very importantly, because that's what's used in prion labs, have worked quite well. We tried that in different cases, and autoclave and formic acid worked quite well in both ways. And one thing we noticed is by doing boiling, we actually had some sort of a precipitate forming in our samples. So we sonicated this precipitate to see if we had a rescue of that, and indeed we had a rescue of that, which basically means that by boiling it, rather than destroying the seeds, we just like clustered them, and then they didn't work on our assay anymore. But the samples were still like seeds were still present in the samples. And then we did high molecular weight rather than brain homogenate, and you can see that boiling actually doesn't work at all on high molecular weight samples, showing that basically really only autoclaving of formic acid works to remove seeds. And then we compared some sort of different samples, because not all lab use brain homogenate the way we do our high molecular weight tau. A bunch of lab use something called sarcosyl insoluble tau, which is, for instance, what's used to show these cryo-EM images that I showed at the very beginning of this talk. And we compared high molecular weight versus sarcosyl, and they are very, very similar, basically, in terms of autoclaving and formic acid work. The only thing we could see is that actually autoclaving in some of these cases didn't work fully, especially in the high seeding cases compared to the low. And that was also the case in transgenic animals where autoclaving didn't work as well as it would on like low seeders. So a couple of take-home message for this talk is that, as I said at the beginning, genetic background has an influence on the amount of tau cells that transfer. Then we saw that tau seeding was heterogeneous from case to case, and that it correlates with some clinical markers of dementia, but also with some biochemical and histological markers. I don't show histological, but trust me on that. And this heterogeneity within AD led us to find out that there were some different biochemical strains that seem to be present and that influence the seeding capacity. And the last bit of information on the destruction of tau seeds, you know, just mine the tau basically. And I just want to open as a last slide to some sort of perspective here, and some sort of questions that remains open on that subject is tau seems to transfer, you know, physiologically in addition to in pathology, and I think we didn't quite understand yet what's the function of this physiological transfer, what tau does at the synapse, and why is it coming, such as, you know, it's quite a big amount of tau that is secreted by cells in a physiological way, and then what goes wrong in tauopathies, why at some point you start to have you know, that this transfer is basically getting bad for the cells. Then, you know, I think all the mechanisms that I talked about or that were shown in the literature are here. Interestingly, these days we're talking about this endosomal escape that is important because tau seeds usually are showing up in the lysosomal pathway, and they somehow are able to escape this pathway and contaminate the cells. And that's like a hot point of work right now on this. And then you have the complex relationship between, you know, like protein structures and all of these cellular mechanisms that are in the brain. Of course, neurons are not alone in the brain. There are a lot of glial cells interactions and complex inflammation or clearance happening here that we quite not understand well yet. I want to thank everybody that was involved in this project, especially these amazing people, Caitlin, Annalise, Tarun, Aurélien, Naomi, and Mark, that have been working in my group for you know, past 6 years. And you know, thank you so much for your attention, and let me know if you have questions to answer them.
Thank you very much for this incredible talk. If there are any questions, feel free to write them in the Q&A. We still have 15 minutes for questions. In the meantime, if I can ask you a question, I was very curious when you characterized the low seeder and the high seeder brains of these AD individuals. I was wondering, like, if you isolated also specifically the synapses, like the synaptosomal fraction, to see whether there was differences in tau abundance at the synapses. And my second question would be, as a follow-up, did you also test different conformational tau antibodies to see earlier stage of oligomeric tau, like T22?
Yeah, so I didn't show that here, but we actually did studies with antibodies to try to reduce tau seeding from these different patients using a quite big range of antibodies spanning the proteins. So we didn't use conformation antibodies, unfortunately, on that one. We used phosphoepitopes antibodies, and also some like pan-tau, more N-terminus or C-terminus. So bottom line of that is that every patient have a different way of reacting to different epitopes that we put. Of course, some of these epitopes are better for most of the patients, and some of the epitopes are not very efficient for all of these patients. But yeah, we didn't try confirmation antibodies on that particular study. The first question was about
isolating the synapses, like doing the synaptosomal fractionation.
Yeah, so we actually did that in a study, not on that one, so not patient to patient, and that's actually a good point and something we could do. We did it in a study to show that basically the presence of tau seeding inside synaptoneurosomes was preceding the presence of neurofibrillary tangles. So that's a study that has been led by Sarah DeVos. At the beginning, I was at MGH, and so yeah, it was very interesting. So it would be possible to do that. But the problem with that is that you have to reserve a bunch of tissue from a lot of different regions, and for that particular cohort, it was not possible to do it. So, but yeah, that would be fairly interesting to do for sure.
Thank you. We have a question in the Q&A. I'm gonna read it for you. It's from Joe. Did you see a role for binding to HSPG in tau seeding or propagation?
Yeah, so not me, but there is the group of Mark Diamond in is it UT Southwestern, I think.
Yes.
Showed that tau, well, basically extracellular tau binds to HSPGs. That's a paper from Holmes, I think, Brandon Holmes in 2014 in PNAS, I think.
I think so. Yeah, and the Diamond group, yes.
And so yes, there is a role of HSPGs. Quite difficult. So that there is also a role of LRP1 into tau integration. So my guess, and I don't know because I haven't done that studies, but my guess is that HSPGs and LRP1 are working together to integrate these tau seeds. Although the problem is that different teams are using different sort of tau preparation, and I'm pretty sure that this influences a lot what tau binds on the surface of the cells and how tau integrates. And if tau is in the extracellular vesicles, that very likely also differs from mechanism of entry to the cell. But bottom line is yes, there is some evidence that HSPGs are involved in that system.
Thank you. So I will take the occasion to add another question. Did you consider checking in these brains across like a hybrid seeds? Like was something that you consider, like to see like amyloid and tau cross-seeded strains, and whether this maybe could lead up to more like an investigation of the different spreading of these hybrid seeds as well, along with the homogeneous seeds?
Saying that we didn't consider is not true, but we didn't do it. We definitely consider it. Yeah, it's always, the problem is like, it's always difficult to find the absolute right model. And these experiments are quite variable already, to be honest, in vivo. So if you add complexity, then you might run into a problem of having too much variation that your whole cohort, you can trash all your data. So that's why we didn't really do it. But that being said, there are some evidence in the literature that amyloid basically have an influence in this type of propagation and increases the spreading of the. I don't remember if I've seen any experiments on seeding per se. It was usually cell to transfer. I might be wrong, but
I think some biophysical studies just demonstrated the existence of these hybrids. That's why I was asking.
Yeah, maybe Margita have done something like that. But yeah, I mean, it's always very interesting to understand how because basically what happens is that amyloid ENTA develops in very different brain regions, and suddenly they kind of like meet in the cortex, and that's where everything goes wrong for the brain. And that's a very simplistic way of seeing this, but there is clearly something that could happen where, you know, tau is doing its own thing and amyloid is doing its own thing, and suddenly when they meet, everything goes wrong. I do believe that for sure there's something there.
Thank you. Is there any more questions? We will just wait for a few more minutes.
Mm-hmm.
Okay, so I think we can close our meeting. Thank you very much for this amazing presentation. And we will be mailing the link of the recorded presentation later. And feel free to reach out to us if you want to ask separately more questions. Thank you.
Thank you so much, everybody.
Goodbye. Thank you.
Goodbye.